Great. All right. It's a great pleasure for me to introduce Lior Pachter, who probably doesn't need any introduction. Lior, you have like 40,000 followers on that former site. and Lior, this is our second keynote, and as you might imagine, we use the star power of Lior to keep you all here, and I think it worked. So all of you are here, and thank you for staying. I tried to figure out whether I had any embarrassing pictures, embarrassing story to tell about you, Lior, but I have none, except for the fact that when I first got to know Lior 20 years ago, because I was working in mathematical phylogenetics at that time, he was at Berkeley. And Aaron, all great single-cell biologists, as you can see from today, leave Berkeley. But as I mentioned, Lior started out in mathematics, and he has gone through the entire journey of genomics, and I believe he now has a wet lab as well. And so really, amongst all the people I know in single-cell biology area, there's really less than a handful of people who really span from deep theoretical knowledge all the way to applications, developing key applications that we use in genomics as well as the now biological experimentation. And Lior is truly one of those people who span all of these areas. But I think more than the contributions he had also made into fundamental science, Lior is also somebody who tirelessly advocates for doing the right thing in science. And this has been really one of the most inspiring things that since 20 years that I have known Lior. Many of us also think that we're trying to do the right thing, but there's, again, only a handful of people like Lior who do it at great personal cost. And so with all of that, thank you, Lior, for being with us. And
thank you so much. Thank you so much, and thank you to all the organizers for having me here, inviting me today. My apologies, I prepared a talk slightly longer than my allotted time, so I may rudely go over a little bit. All right, so today I'd like to talk about the systems biology of a cell and explain a little bit what that means. So just like to begin by recalling the history of this meeting. There's actually a very nice article written by Sten Linnerson exactly 10 years ago when this meeting was young. Meeting, I think, launched in 2009. But by 2013, it was really a thing. And if you read this article, one of the things Sten points out is that, the attendance was very strong, and this young field is here to stay. I think that's been borne out by coming to this meeting, which has been exceptionally interesting and really fun. It was also clear that, there were challenges ahead, but we have a field. I think that's true. Sten ended his piece making really two predictions. That with more accurate measurements comes the ability to study subtle quantitative processes, such as transcriptional noise, and with large cell numbers, the ability to study intercellular heterogeneity. And these are wise words. They're good predictions. One of them has been borne out, the intercellular heterogeneity, and I'm going to argue to you today that the other one really not so much, and I'd like to talk a little bit about why and whether we should do anything about it. That's what my talk is about. So let's start with intercellular heterogeneity. There's actually dozens of articles with that in their title, and I just pulled up two here. I think it's fair to say almost every single cell genomics paper, not all, but almost all, are about characterizing intercellular heterogeneity, and Sten was spot on. I myself began in this field essentially working on determining intercellular heterogeneity. It's very, very interesting. This was one of my very first papers when I moved to Caltech about 7 years ago. I had the great opportunity to collaborate with David Anderson. He's a well-known neuroscientist at Caltech. He works on the VMH, it's the ventromedial hypothalamus. It controls social behavior. And in analysis in mice, with my student Lin Yi and his student Dongwoo Kim, actually found a female-specific cluster. This kind of thing is really fun to do, and that's kind of how I got started out. We actually had data from SmartSeq as well as 10X, and that played a very minor role in this project, but it was to try to integrate these technical modalities. A few years later, I worked with a former student of mine now, Sina Buisagi, who's in the Streets lab now. But we looked for isoform heterogeneity. So we were looking at the brain and part of a big consortium to study the mouse brain, starting with the primary motor cortex. And we did a similar thing where we looked for, in this case, clusters that are separated by virtue of isoform markers. So there's sex-based intercellular heterogeneity, isoform-based, with another student who spoke here a few days ago, Tara Chari. We, in our lab, did single-cell RNA-seq on this tiny jellyfish. I'm pretty proud. I think it's the first single-cell RNA-seq atlas of a jellyfish. Again, our goal was intercellular heterogeneity. In this case, we were actually interested in neural cells. The jellyfish has a kind of actual neural network, like a real neural network. So that's how I got started in this field. I think it's fair to say intercellular heterogeneity has been a win. Valentine Svensson has a database. It was shown I think a day or two ago. He keeps maintaining it. I just reran it. These are the number of studies and the number of cells in them. There's a recent study of the developing mouse embryo from the Shindura lab with 12 million cells. One of the interesting things is the more cells you sequence, the more heterogeneity you find. And whether that will continue forever remains to be seen. Eventually, every single cell will be its own cluster with some interesting biology. All right. But what about subtle quantitative processes? So first I want to discuss briefly what did Sten Linnerson mean when he said subtle quantitative processes. And to understand that, we need to go back a little bit earlier in this meeting, and I got to borrow this photo of Sunny Shi from one of the very first meetings here. And we heard on Thursday morning it was a tiny meeting in a little room. And Sunny Shi was one of the, I think, sort of pioneers in single-cell biology, which precedes single-cell genomics. And he studied... this is done with fluorescence imaging. Transcription and translation by virtue of just imaging single molecules and looking at what's happening. And really, the point of this article, it was co-authored with my current colleague, Long Kai, is that transcription happens in bursts. It's kind of, there's stochasticity to expression, and it happens in bursts. And studying this quantitatively is what they did in this paper and several others at that time. There's actually one equation in this paper. It's this equation. And it's describing in steady state, under some stochastic process that I'll say more about in a minute, how many molecules you'll have. It is a continuous model rather than talking about molecules, because what the measurements at the time were intensities. It's sort of, a dynamical, continuous model. And I just wanted to note here that in this article it's explained that, this, whatever this thing is consistent with a negative binomial distribution in some discrete version of this. I'm going to show you the discrete version of this today because that's what we actually are interested in now and is relevant now, because we measure molecule counts when we do single-cell RNA-seq and not intensities necessarily. Okay, so what kind of interesting subtle quantitative processes did they study and discover? Well, for example, the Fano factor is sort of, like the coefficient of variation, the variance over the mean. They looked at this and actually measured that it's different from 1, implying that these processes are not Poisson. That's interesting. That tells you something about the stochasticity of transcription in a cell. It's got nothing to do with technical noise. In the same article, which, by the way, was published in Science in 2010, there's an interesting result about the rates of transcription versus translation being uncorrelated with each other. In fact, that's sort of the main, it's highlighted at the top. Devil in the detail. Remarkably, mRNA and protein levels are uncorrelated. This is actually a little deeper than that. A and B here are rates in this dynamical system where DNA is transcribed to RNA, degraded, and then translation happens. And so in this equation, these terms essentially correspond with rates that are being measured. And there's some interesting understanding here of these processes and how they relate to each other. Okay? That's what Sten Linnerson was talking about. But none of single-cell genomics actually, or almost none, I should say, a little bit, but almost none, is quantitative today. Now, how do I know? So that's what I'm going to tell you, all right? So the reason I know is because all of our analyses start with this transformation. X goes to log x plus 1. I'm going to explain to you what this transformation is. And then you'll see that it means that nothing we do anymore is quantitative. And I think that's a shame. So I'm here to advocate for putting the quantitative back in this meeting and in this field. All right? So how do I know? Well, let's break this apart. This is a pretty, it's the second equation I'm showing. It's pretty complicated. So let's start with x goes to x plus 1. Here at Cold Spring Harbor, I know this is understood because every morning I'm staying up here in Dolan. It's like up the steps. At 7 a.m., the bell tower, which is right next to us up here, rings 8 times, okay? So that's how I know that here we understand this transformation. All right. Then the logarithm. The logarithm is what you do, when you can't have enough paper So it shrinks things down. That's what the logarithm is. All right. Where does this transformation actually come from? It comes from actually statisticians in the 1940s, in particular Anscombe is a well-known statistician, who showed that if you have data that's distributed according to the negative binomial distribution, and I don't have time today to talk much about it, but our single-cell RNA-seq counts look like that, and that's well known. These are kind of one of the exploratory plots you might make here, where every dot is a gene sampled across many cells. This is the mean across the cells and the variance, and you see plots that are not linear. That would imply some Poisson sampling, but that tweak up a little bit, and that's because possibly there's just additional technical noise. This is very common with count data. This relationship between mean and variance is not good if you're going to do the next steps, which are the standard analysis like PCA and all sorts of other analyses. Because essentially, if you don't adjust this relationship, then it's only going you're going to weight towards highly variable genes. Essentially, only your analysis will ignore all the lowly expressed stuff, for example, transcription factors often. So Anscombe figured out a way to transform data like this. So the data is X, Y, these are constants. Now, recently, actually, I have some kids, and there's a lot of debates about whether they should learn calculus. And I'm going to be honest with you. I don't remember much calculus. And I learned this in calculus, and I had to look this up because it's the inverse hyperbolic sine, and most people don't know what that is. But you're okay. It's a simple thing. It's well approximated by the logarithm. And this transformation stabilizes the variance. It basically, if you apply this transformation, your genes will have roughly equal variance after you apply this transformation. Now, that's a reason why all analyses essentially begin by this log x plus 1. Why 1? Well, really, the pseudo count here should match how steeply this curve is going up. It's related to that slope or derivative, quadratic coefficient here. I think people put 1 because, I don't know, it's a small integer, okay. But the thing is, I just showed you that Sunny Shi's mindset, the folks in systems biology studying single molecules by fluorescence understood that the negative binomial distribution arises from intrinsic stochasticity in the cell. It has nothing to do with sampling. And so the very first step in all of our analyses is to throw out all the information that would facilitate understanding that very process. We just flatten out the variance, we're done. So that's maybe a shame. Maybe we shouldn't start with throwing out all of this stochasticity. Well, now, the situation is a little bit worse than that, okay? My student, Gennady Gorin, who just graduated, did the following thought experiment, which is to say, suppose we have 3 cell types, and we have lots of cells in each. We can estimate the mean expression for a gene in each of them quite accurately, actually, because we have lots and lots of cells in each cell. And we can also get an estimate of the variance, maybe not as accurately, but we could use more than 3 as well. And so what you can assume is that the technical variation, to the extent that it exists, is averaged out when you look at the mean within a cell type, and the variance between cell types is all biological, right? Maybe also, maybe a little bit technical for some cell types are better sampled than others, but there's biological variation there. And so you can ask, with that as a bound, what is the transformation doing to the variance? So we made plots like this, and I'm short on time today, so you would like to be in the green region after your transformation, okay? So if you drop out of the green region, you've violated this lower bound. By normalizing, you've removed so much variance that you've lost the biological variability between cell types, let alone the stochasticity of transcription that's inherent to cells, that too, but even more, okay? So here's what it looks like in the standard analysis. So the first step, we call this proportional fitting. This is when people depth normalize each cell to have the same number of counts. It's actually the standard normalization is to do that. That's the total number of counts per cell. This is the counts per gene per cell. So it's not just the raw counts. Some cells have more counts than others. First, that's normalized out. That also throws out biology. In fact, you can see that some of the genes are a little bit above this line. That means you've added noise. When you do the logarithm, you suppress the variance. You're variance stabilizing. Everything's getting the same variance. So all of these red dots, which are the most highly expressed genes, have now had some of the biological variation between cell types removed. That's not great. Then people do PCA, and that removes more variance. And finally, UMAP actually injects noise back in. So it's going up here. More points are above the green line, but that's really bad because you've lost the biology, and now you just inject noise. So that's really awful. Now, I'm a bearer of bad news, but I'll get to the good news in a second. Just bear with me, okay? So the gold standard today for analysis is 2 amazing programs, Seurat and ScanPy. I think they've really transformed the field in terms of looking for intercellular heterogeneity. They're the standard workflow. Well, one of the things they do is they implement the logarithm function. It's like very pretty simple stuff, like log x plus 1. And last year, I learned via Twitter, actually, about a paper by Davis McCarthy. I learned this from Nikolai Savlov, that they were different in their log-fold change calculations. So we took a look. This is my student Lambda Moses and a new rotation student, Joe Rich. This is Seurat versus ScanPy. This is the log-fold change between 2 cell types or 2 clusters in an experiment. It's a 10x experiment. They're implementing log A minus log B. You would like to just be on this gray line, but this is what the difference looks like when you calculate fold change with Seurat versus ScanPy. I'd just like to point to your attention, some of the points here are off by, this is log, okay? All right? This is not good. This should make you a little bit uncomfortable. And that's what the distribution looks like. Well, why is this happening? This isn't math, this is arithmetic. Actually, neither of these software packages have ever published properly these formulas. We found these formulas by reading the code, and they're both a little wrong. The Seurat people have now corrected their formula. I won't get into the details in Seurat version 5, but I will note that there's been several thousand, 10,000, 20,000 papers that have used Seurat 1 through 4. And so, I don't know, maybe it doesn't matter. Okay. Maybe. All right. Okay, so this is in a supplement of our paper. All right. Now, why is this? Well, why are they not even publishing their formulas? I don't blame them. In 2012, there was an article that use of equations impedes communication among biologists. And I would say not even light use is really tolerated. just as a little exercise, I walked around the posters yesterday, and I counted how many had an equation on them. And, not to offend anybody, but I counted 4 posters out of around 50 had an equation on them. Almost everyone had a UMAP. We'll get to that in a second. All right. Okay. So this is not new. In fact, I gave a talk right here at this platform almost 10 years ago to the day at Genome Informatics, which was in early November 2013. It was titled Stories from the Supplement, and I complained that equations are not published in articles. So that was the thesis of my talk, and and really nothing has changed. In fact, I haven't changed. I'm wearing the exact same clothes. Okay. All right. Okay. All right. So you might say this talk today is Stories from the Supplement Part 2, Tales from the Cell. All right, that's what we're... All right. But here's some good news. So we did a little experiment, and we asked the question, okay, Seurat and ScanPy are different. Let's say we don't really care. Well, we might as well not do so much sequencing. So this green curve is the difference in log fold change you get if you downsample the data to 2%. So if you really don't care about this log fold change, and maybe it doesn't matter for the final result of your paper, then I'm here to tell you the good news that you can save 98% of your sequencing money. Don't bother. There's no need to sequence, all right? Okay. And even better, if you're going to just focus on your UMAP and clustering, you can sequence even less than that. Maybe 1%, 0.5%. And why do I say that, actually? So with Sina Buishagi, we wrote an article last year where we reverse engineered the Seurat. Seurat actually keeps around several count matrices simultaneously that are some are log transformed, some are not. They're used in different ways, in different functions. The UMAP first actually runs a program called Annoy. That makes something called the K-nearest neighbor graph. Yeah, I didn't make these names up. I'm just here to tell you, okay. Actually, I think something rather fundamental that many people don't realize, so I thought I'd mention it to you, is that this same K-nearest neighbor graph is used to cluster. So actually, when you look at a UMAP, it's trying to not visualize the cells, but actually it's trying to visualize the K-nearest neighbor graph. And the clustering methods, which are based on these Louvain, Leiden, these methods, community detection methods, are also their input is the K-nearest neighbor graph. So what you're actually looking at is the K-nearest neighbor graph 2 times. And but I think a lot of people look at the UMAP painted by the clusters and go, okay, the UMAP worked, like the clusters are separate. But that's by design of how it works. It's not really an independent clustering. Well, why does this matter? We actually looked at whether Seurat and ScanPy build the same K-nearest neighbor graph on the same input. And this is just the degrees. You can focus on this corner here. This is Seurat versus ScanPy. They don't build the same graphs. So you can actually sequence even less. If you really don't care whether you use Seurat or ScanPy, then sequence less. Okay, that's great news. All right. I don't have time today, but this was mentioned. My student Tara Chari also wrote a whole paper about UMAP. Even if the K-nearest neighbor graphs were the same, and even if the log was computed the same way, and the PCA, which are also different between these software packages, there's many, many problems, and those I won't talk about. So let's talk about what we should do. And so I'd like to spend the remaining time talking about what would constitute a systems biology approach in the spirit of Sunny Shi and some of the other pioneers in this area, which I think is eminently tractable and approachable today with the data we have. The observation is, first of all, that the negative binomial distribution arises as the steady state for the bursty model. This is the primary accepted model. There's an insight that is not due to us, but actually also thanks to Sten Linnerson and his group, that in standard experiments, we actually have access to estimating parameters for bursty models and other related models, because even just standard vanilla single-cell RNA-seq experiments are actually multimodal, and I'll explain what I mean by that in a minute. I'm going to talk briefly about software we've written in our lab to perform these estimates, and then I won't have much time or any time to talk about details, but I'll just survey some of the work by my students over the last 3 or 4 years, really developing this theme into all of the analysis aspects of single-cell RNA-seq. Okay, so let me just start very briefly with discussing the models. The most basic modeling for really single cell biology goes back to the 1950s, thinking about transcription as a dynamical system where there's some production rate of molecules and some degradation. X appears here, right, because the more molecules you have, the more will degrade, because any one of them can degrade independently. It's actually due to Jacques Monod, and in fact there's a beautiful I think illustration of Jacques Monod downstairs in Blackford Hall. So the thing about a model like this, it's a very, very simple differential equation, is it's deterministic. You start with some initial condition, you always get the same happening. That's not how cells look. So there's many, many experiments being done, mostly by fluorescence imaging, tracking transcription over time and noticing that it's bumpy, okay? It's constantly changing. It's the stochasticity in cells. One of my other colleagues at Caltech, Michael Elowitz, in the beginning of his career, wrote a really beautiful, I think, classic paper showing how you can tease apart by an interesting experimental design inter versus intracellular noise. And okay, so we should be able to recapitulate that and more today. So how do you fix the basic ODE? Well, you introduce what's called Brownian motion. So Brownian motion is essentially a random process that adds stochasticity. It's the simplest way you can do that, and goes back to Robert Brown and actually Einstein, who essentially published the math of Brownian motion along with just a handful of other small papers in 1905. Okay, but there's a problem. So so what that leads to in math is a differential equation like the one I showed before, but with a new term called epsilon, and there's a lot buried in here, too much for today. It turns this equation into what's called a stochastic differential equation. There's now randomness in the differential equation, and this is a field of math that's used to study such equations. The problem with this equation is that, it's great now, like this is simulating from this equation. This is the mean. It looks like the ordinary differential equation because the noise is zero on average. But sometimes you bounce down to negative numbers of molecules, which is not good. And this has been fixed. These models are studied in mathematical finance. There's a famous model called the Cox-Ingersoll-Ross model. It's used for interest rates because, well, in the good old days, they maybe didn't go negative too often. We'll see what happens in the next few years. But essentially, by adding this term over here, this square root of x, you essentially dampen the random motion as you get close to zero, prevents you from going down. The only issue with this model is that the x here is a real number, but what we actually are measuring with UMIs are molecule counts, right? Our data is discrete. The way to fix this is with something called the chemical master equation, and this is a model from actually mostly popular in chemical engineering, and I'm sorry my slides keep vanishing. So this model discretizes that random stochastic differential equation. The way this works basically is you have a Markov chain. This is a very simple example where there's production and degradation. You might have 0 molecules, 1 molecule, 2 molecules. I like to imagine these as knots on a rope. And you're walking up and down the rope. And there's a probability of jumping up in the rope, that's production, or jumping down when one of the molecules degraded. The further you go up the rope, the more likely it is you jump back down because, again, every molecule can independently degrade. One of the features of this model is you can't jump to -1 molecules. This model is interesting because the state space is discrete, but time is continuous. Okay, so there's at any instant a probability of one of these jumps happening. So it's a continuous discrete model. This model is called the constitutive production model. And I won't prove it today, but its steady state is Poisson distributed. So having this kind of stochasticity underlying your transcription will actually lead to Poisson distributed data. And a small tweak on this model is called the bursty model. It's super simple. The production part now has another switch attached to it. If it's off that switch, nothing happens. And when it's on, you're in this model, okay? And there's just there are now new rates of switching between off and on. So in this model, it adds more noise. You can see that because there's being often on adds stochasticity. And this model, in its steady state, generates negative binomial distributed data. That is what Sanishi was referring to in that paper in 2006. Okay? Now, I often, when I've been talking about this recently, people say to me, are you serious? You're going to try to model your data with this kind of crude model? And I'll tell you that it is a terrible model. Its purpose is not really to fit the data, but to sharpen the questions, okay? What we can do when we start modeling with this data with this model, we can see if the data fits. We can see what aspects of this model need to maybe be changed or tweaked. So we start simple and get more complex. I will also tell you that the model you are using right now, when you run the standard workflow of log, PCA, UMAP, it's a worse model. It's much, much simpler than this one, okay? And we can talk about what it is. It's a really bad model. So this is less bad, okay? All right. So I don't have much time today to go into all of the details of this. My student, Gennady Gorin, he just graduated, as I said, this past June, really had the vision to sort of analyze the single-cell genomics data this way. We have recently published a paper in Cell Systems, and it has a lot of equations. I'm very happy with that. It's together with John Vastola, who was an applied math student at Vanderbilt when we did this, who's now a postdoc at Harvard. And we explain an entire theoretical setup via generating functions, how you can write down fairly complex models that capture the entire technical aspect of generating just single-cell RNA-seq here, and how you can do inference. Our models can include noise parameters for the actual technical artifact, not just for the stochasticity that I just discussed. That's biological. And the reason we're able to do this, okay, is because of this insight from a paper by Lamano, this is Sten Linnerson's group, introducing RNA velocity in 2018. They had a very interesting insight, which is that with 10x data, although it's even better with other kinds of single-cell data, you can actually generate two matrices, what we call the spliced and unspliced count matrices. These consist of counts of reads that map to introns, which presumably are some kind of nascent RNAs that have not yet been post-transcriptionally spliced and processed, versus the spliced reads. These might be reads literally crossing junctions, or maybe you would include exons that are ambiguous, but more likely to be processed from processed transcripts. And these two matrices essentially give you 2 time points in one snapshot experiment, because these counts are sort of predictor of what the RNA will look like, the processed RNA, in a couple of minutes. So their idea was to make phase diagrams like this, where if you have higher unspliced counts, your pump is primed, and you're about to see an increase in expression of your gene, versus the other setting where your unspliced is weaker and you have a lot of spliced counts. It looks like you're done transcribing, you will degrade your molecules, and your gene is shutting off. So you can predict the future, and using that, they put arrows on the UMAP dots, pointing by averaging over lots of genes as to where you're going. So it's a very interesting idea. I will point out, and I don't think it's been thought about too much, that the moment you understand that there's actually two matrices, in any analysis you do, whether it's really sophisticated deep learning or some manifolds, or whether you're just looking for clustering, you should ask yourself, which of these two matrices should I use? And there's actually several options. You could combine them, sort of paste one underneath another. You could average them. You could pick one or the other. You're going to get different answers. Tara, I'll mention in a second, has studied that for clustering. Any analysis you do, you're faced with this dilemma, and if you choose to ignore it, you should be asked when you give your talk, why? Okay, so that's a good question. Now, RNA velocity is appealing at a conceptual level, but the implementation of it was, in my opinion, flawed because it involved lots of heuristics, and together with Tara and another student, Mei-Shen Fang and Gennady, we wrote a paper, and it's in PLOS Computational Biology just last year. We analyzed the entire process because there's not just, this is just intuition, but you actually have to have a model. Which model did they use or should one use is a question, but there's also processing step, this log I just mentioned, this k-nearest neighbor graph. And it turns out that, well, there's different packages. We've seen SC Velo used at this meeting. There's the original Velocyto. In this example from the original paper, these are differentiating neural stem cells. It looks like a beautiful picture, but here the arrows go the wrong way. And it's a bad habit I have, but I like to look at all of these pictures, so we looked at lots of papers, and many of them have arrows going the wrong way. And it's actually so many parameters in the method, you can kind of make the arrows go wherever you think they should go. And so it's nice to make pretty pictures, but it's not a quantitative method in the sense that Sten Linnerson was writing about 10 years ago at this meeting. So I'm going to wrap up soon. I'll tell you a couple of things we're doing in my lab without giving you the details, which I apologize for, but just to give you a sense. So 2 of my students, Delaney Sullivan and Christian Hjörlefsen, have redeveloped our processing program, Callisto, to accurately estimate these counts, which is, in fact, rather technical to do exactly, and we care about the numbers, so we have a paper about how to do that. We've also incorporated several improvements to related programs, one's called Bus Tools, and some other work that has been done by Sina Buesagi. There's some work about translated mapping by Delaney and another student, Laura Lubert, so we have a release of Callisto pending shortly after this meeting. Gennady implemented the inference in a program called Monod, named after Monod, for inferring the parameters in these models. I don't have time to talk about it today, but it's a really, really cool piece of software because I think it's the first time that we have a tool that can take an actual single-cell RNA-seq data set and estimate these parameters. Now, I just want to be clear, Monod estimates parameters for bursting frequency and burst size, degradation, transcription production per gene. Because if I go back just to these phase diagrams, we need the whole phase diagram to get one set of parameters, okay? So these are not cell-specific estimates, but they're gene estimates that are using all of those cells that we've sampled, okay, in this quantitative way. First of all, we can check, and Monod actually has a noise part of its model for technical noise, but we can actually check whether Monod is removing variance, and I don't have time to explain the whole plot, but the answer is no, we are not violating the bound across cell types. So we might be harming our data a little bit, but not too much. We have access through these parameter estimates to a whole new kind of analysis. Because whereas before, this is from the same brain data set I talked about in the beginning of the talk, primary motor cortex. This is just looking at GABAergic and glutamatergic cells, a very basic 2 cell types in the brain, inhibitory and excitatory neurons. We can look at burst size fold change. Now, the thing is that usually what people do is they look at the count differences, right? What we really have done is we've integrated the 2 modalities, the unspliced and the spliced count matrices, to have access to multiple parameters, okay? Not just one number that we're comparing between 2 cell types. And so this is showing that we can find differences that are under the hood that you would be blind to just looking at spliced count differences. And we're often asked about validating this. We've been working very hard to do that, and we have some decent initial validations, with, especially with spatial genomics data, where we have fluorescent in situ hybridization as a sort of alternate way to measure expression. I don't have time to go into that, but I think it's very exciting because we can take any data set that you all have already published and look at it from this new point of view. Couple of remarks. Doing the math for Monod is complicated. We're solving not-so-easy equations, and we have a really nice paper led by another student in my lab, Maria Carilli, where we show how neural networks can be used to solve the math. And this is in the theme of a really cool body of work where neural networks are now used to solve PDEs, for fluids especially. You can use neural networks to solve the chemical master equation. We focused on this application, and it involves some interesting ideas, but I think they're generally useful for these class of models. Maria, in another paper, has, together also with Gennady and Tara and Jankin, who's a student at UC Davis, adapted SCVI, which is an autoencoder framework, to place this biophysical model in the latent space. And it's really, really a cool idea, because what it allows you to do is interpolate these parameters we estimate per gene with Monod into single cells. So you can get a cell-specific parameter estimate. And anyway, there's a preprint, and it's currently under review. I will just show the slide. What Tara has worked on is a clustering approach that is based on clustering, not on counts, but on these parameters. And it solves a really vexing sort of problem, which is that I told you there's these two count matrices, these two modalities in any experiment. This provides a biophysically motivated and coherent way to integrate those modalities for clustering. So her clustering approach is based on clustering on these biophysical parameters instead of just one matrix or the other. It's a really beautiful work. I don't have time to get into it, but we show that because it's model-based, we can actually estimate the number of clusters in a coherent way, and we don't run afoul of artifacts that come up when you use one matrix versus another. One or two more points to make. There's a field of trajectory inference that is, I think, very interesting, where people try to order cells. But again, you take this count matrix. Well, which count matrix should you use? So Meishen in my lab, and this has been a project that's been 2 years of a lot of work to figure out how to do this, has thought about how to turn pseudotime into process time, where T is actually the parameter in a model that for transcriptional dynamics. Again, I don't have time to talk about it. This is not quite ready, but we'll have a preprint out, I think, in the next few weeks explaining her method. And one thing I really find pleasing about this approach is that it interpolates naturally between clustering, in the sense that I just talked about that Tara has worked on, and trajectory inference. In fact, we have a model-based approach to each, so we can take a data set and ask, does it look like the cells are clustered separately, or does it look like there's a trajectory? And we have some very nice control experiments where you just stick data into current trajectory inference methods. They will always find a path, whether or not it's really there. Final two extensions we work on, and then I'll be done, is Kayla Jackson in my lab is focusing on how to model spatial data from, again, the subtle quantitative process point of view. And Catherine Fels is looking at chromatin accessibility data, ATAC-seq, how do you incorporate such data into this kind of modeling, and how to think about gene interactions, which are, of course, super important and very, very interesting and very complicated. So I'll just stop there. I don't have any funders listed here because nobody funds this kind of work, so sorry. Okay. I'd like to show my whole lab. Almost everyone is here, not everyone, but Gennady sort of had the vision for this kind of program of research, but really a lot of the lab or almost all of the lab is engaged now in thinking about these models and how to think about the data, and it's a lot of thinking and not always much doing, and it's really fun, and it's hard to credit every person individually because we just brainstorm as a team, and it's really been a pleasure. So I hope that we can help to bring back some of the quantitative into this meeting. Thank you very much.
I think we have time for some questions.
That was an excellent talk. Thank you very much. So I'm just wondering, in terms of clustering based on your inferred parameters for your biophysical models, so when we look at our RNA transcripts during the cell cycle phase, we see changes in RNA abundance depending on which phase the cell is in. And so if you're analyzing cells that are proliferating, how would you advise to go about measuring those parameters and performing the dimensional reduction based on those parameters?
So there's several aspects to your question. First of all, these models assume a steady state. It's a weakness in the modeling, but in Meishen's work, we have a sort of piecewise constant parameter approach to thinking about cells that are proliferating or differentiating. In terms of cell cycle, there's definitely genes that are cycling and are going to not be modeled well. I think there's also arguments that many genes, have bursting that's not based on a sort of simple on-off transcription factor simplistic model. So we don't have good models for the real thing yet, actually, in a way. But again, the alternative is worse, because if you're just taking log and using the count matrix, you also are having a model where you're also ignoring that and more. So I don't have a good answer for you other than that. We try to sharpen the questions with what we have. Yeah,
so perhaps sort cells based on G1 phase or something.
Yeah, but we don't separate out right now in terms of the cell cycle phase.
I have okay, Nima from Sanofi. A very nice work, actually. The type of humor that you used here, well received. I think it's really lack the grant for the specific type of the job you are doing. I think we are missing this kind of going inside of all of these packages and looking. I really appreciate it. You showed two equations that was mistaken, and you mentioned that it was corrected. I wanted to make sure that what was the mistake and what was the correction.
Okay. Yeah, I'll just say briefly. Sorry. Yeah, you're asking about, where did I go? I'm sorry, I'll be quick. So you were asking about these equations, I think, is that correct? Yeah. Yeah, so the problem, I'll talk about Seurat first. I think when you do a logarithm of a number, one reason to add a pseudocount is that if you put in 0, you get an undefined number in the log. So in Seurat, what's going on here, the y's are the logged numbers, log x plus 1. When you exponentiate that and subtract 1, you get back to x. And then they've added a 1 because they're going to take a log. But it's a very large pseudocount if your average expression in a cell for a gene is very low. And so for very low expressed genes, this is completely off. That's why in Python, in ScanPy, epsilon here, their pseudocount, instead of 1, they use 10 to the negative 29. Okay, so it's a mistake to use 1. It massively distorts fold change on low expressed genes. That's the problem here. Here, it takes a moment to understand what's happening, but there's a geometric mean, and they're taking the geometric mean of xi plus 1. So, there's x1 plus 1, x2 plus 1, taking the geometric mean and subtracting 1 at the end. And unfortunately, the geometric mean of, that's not a correct thing to do in mathematics. You can't add 1 to every number, take the geometric mean and subtract 1 at the end. That's not the same as the geometric mean of the numbers. So it's a more subtle mistake and less problematic in ScanPy, but yeah.
So Joseph from Vanderbilt University has a question on Zoom, and he says that you mentioned that 2% of sequencing is sufficient for single cell data, and does the rationale behind this account for the detection of rare cell types and transcripts that might help one tell the difference between subclusters of cells?
Okay, that's a great question. Yes, please don't take me too seriously. So, oh my God, yeah, I'll get sued. Yeah, of course, if in fact, for the purpose of having the same difference in log fold change between Seurat and ScanPy, you could go down to 1%. Then you cannot even call anymore what's a real cell. So, yes, of course, there are reasons to sequence deeply to look for rare cell types. But what I'm saying is, for a lot of the kind of analysis we do, which is just to cluster the data, to get a cursory understanding, to find some marker genes, we are massively wasting our money in the sense of not caring about the methods that are being used. The methods really matter. And I think if your sequencing depth is solely to find your rare cell type, you have to ask yourself, how much money is that rare cell type really worth for me, right? Because some of these experiments, when you think about how much sequencing goes into like a 12 million cell, 20 million cell, soon we'll be at 50 million cells, these are not projects that cost $1,000, right? They cost millions of dollars, actually, of sequencing costs. It's great for the sequencing companies, but maybe there's other things you can do with that money.
Hi, fascinating talk. I was wondering, what is your opinion of what do you think we're missing from modeling gene expression as an event, as a process of events, by abstracting from the actual structure? So like secondary RNA structure, do you think we need to pay attention to these things?
It's a very good question. Yeah, so structure really matters. One thing Catherine has been thinking about is open chromatin, and you can think about modeling high C data. You're asking about RNA secondary structure. I don't know. I haven't really thought about it, but absolutely could be very interesting to model, to create very complex models in the future that are sensitive to all of these aspects that we know affect the molecular biology of the cell. That's a very good question.
Hi, Lior. Great talk. Just a couple of questions. The first one is regarding the spliced and unspliced counts. It's my understanding, based on my crude understanding of splicing, that by the time the genes are polyadenylated, you're actually losing a lot of the nascent transcripts. So are these transcripts even a count of nascency, and what do we lose by just assuming they're all mature transcripts?
It's a very good question. Yeah, so that's correct. The way, like in 10x polyadenylated capture works is there's a bead with poly T on the end, and polyadenylated transcripts get grabbed. Those transcripts have been processed, they're mature. But the insight that Lamano and Linnerson had was that there are poly A tracts in introns, just sequences of A's in the genome, and nascent molecules have those A's on them, they get captured. So they get captured before they have been processed, and not because they have been polyadenylated, but just because they contain a stretch of A's. So that is why we have access to that information. Yeah.
And so you think it's an even sampling of those, or there's no bias against any type of nascent molecule?
So there's a bias because, several biases. So first of all, the number of poly A tracts in your gene matters. And so in these models we have there's a length, the length of the introns increases the number of nascent counts you get, or unspliced counts. That is an absolute bias, and we account for that in Monod and these models that we have. With other technologies, that's a good question, but it's a lot I'll just summarize by saying there's single nuclear RNA-seq that has different features. There's full-length RNA-seq. Yeah, everything has different biases and different issues. Also, to do these analyses, we need UMIs. And some technologies, for example, SmartSeq has full-length RNA-seq, but it doesn't have UMIs, at least the initial SmartSeq v2. SmartSeq v3 has some reads that are 5' UMIs, some not. So it's a whole world of interesting things to think about. Yeah, good question.
We'll have one last question from the virtual audience, and then.
So you mentioned some corrections that should be made, etc. And so I was wondering, what do you think is the path forward from here? I mean, do you think editors should take cognizance of this and issue corrections and amendments to the papers? Or, I mean, what do you think should be done to solve these problems?
Okay, what do I think should be done to solve these problems? We have a panel coming up.
Yes, that's right.
Maybe I'll just say, one thing. I recently, maybe I'll very briefly tell in 20 seconds an anecdote to answer your question. I live in Pasadena, California. We have a big mountain, Mount Wilson, behind our campus. And in the 1920s, Michelson measured the speed of light by triangulating the distance between Mount Wilson and Mount Baldy, which is 20-something miles away. And he did it by triangulating with string, accounting for the catenary curves. He got the speed of light to within a few decimal points because he really cared about the measurement. And I think in the physics community, it has its flaws and whatnot, but there's when something is measured, the charge in an electron, the speed of light, gravity, anything, you don't just do it once and move on to the next paper, right? And make a UMAP. You try to do it again and again and really think about the accuracy of your measurement. And I think what I'd like to see is just the community as a whole appreciating and thinking about questions like, I've gotten an RNA-seq data set. What do these numbers really mean? How accurate are they? That involves more controls, more replicates in experiments, just greater attention to the technical detail. It costs money and it costs time, but ultimately it moves you forward to actually understand the phenomenon of interest in a much, much deeper way.
Great. Thanks again, Lior.
Thank you.